Microbiologically induced corrosion, or MIC, damage mechanisms is a part of uniform or localized loss of thickness. Description of microbiologically induced corrosion based on API 571, a form of corrosion caused by living organisms such as bacteria, algae or fungi. It is often associated with the presence of tubercles or slimy organic substances. Microbiologically induced corrosion, or MIC, is a type of corrosion that occurs due to the presence and activity of microorganisms, such as bacteria and fungi, which can accelerate the degradation of metals and other materials. MIC is a significant problem in various industries, including oil and gas, water treatment, food processing, and marine operations, where metal surfaces are often exposed to harsh environments conducive to microbial growth. Description of microbiologically induced corrosion, or MIC, damage. Number 1 is mechanism of corrosion. Number A, of mechanism of corrosion is biofilm formation. The process often begins with the attachment of microorganisms to a surface, forming a biofilm. This biofilm consists of a dense layer of microorganisms encased in a protective matrix of extracellular polymeric substances, or EPS. Number B, of mechanism of corrosion is metabolic activity. As these microorganisms metabolize nutrients available in their environment, they can produce corrosive byproducts such as organic acids, hydrogen sulfide, and other metabolites. This metabolic activity alters the local environment, leading to localized corroding agents that attack the underlying metal. Number C, of mechanism of corrosion is electrochemical reactions. The presence of biofilms can facilitate specific electrochemical reactions that may not occur in their absence, exacerbating the corrosion process. For instance, some bacteria can induce changes in the local pH or create microenvironments that favor corrosion. What is biofilm formation? Biofilm formation refers to the process by which microorganisms, such as bacteria, fungi, and archaea, adhere to surfaces and create a structured, complex community known as a biofilm. This process is characterized by several stages and can occur in a variety of environments, including natural ecosystems, industrial systems, and the human body. Number 1 is stages of biofilm formation. Number A, of stages of biofilm formation is initial attachment. Microorganisms move towards a surface and begin to adhere, often using structures like pili or fimbriae. This initial attachment is usually reversible. Number B, of stages of biofilm formation is irreversible attachment and growth. Once attached, some microbial cells begin to divide and form microcolonies. The adhesion becomes stronger and more permanent at this stage. Number C, of stages of biofilm formation is extracellular matrix production. The microorganisms in the biofilm start to produce an extracellular matrix, composed of polysaccharides, proteins, and nucleic acids. This matrix provides structural support, protects the microbial cells, and facilitates communication between them. Number D, of stages of biofilm formation is maturation. As the biofilm grows, it undergoes further development and maturation. The biofilm can evolve into a complex three-dimensional structure with multiple layers and channels that facilitate nutrient and waste exchange. Number E, of stages of biofilm formation is dispersal. Eventually, some cells may detach from the biofilm and disperse into the environment, where they can colonize new surfaces and form new biofilms. Number 2 is characteristics of biofilms. Number A, of characteristics of biofilms is complexity. Biofilms are composed of diverse microbial communities that may consist of different species, forming a stable ecosystem. Number B, of characteristics of biofilms is resistance. Microorganisms within biofilms often exhibit increased resistance to antibiotics, disinfectants, and environmental stressors compared to their free-living, planktonic, counterparts. This resistance is partly due to the protective nature of the extracellular matrix and the altered microenvironment within the biofilm. Number 3 is implications of biofilm formation. Number A, of implications of biofilm formation is corrosion. In industrial settings, biofilms can contribute to microbiologically induced corrosion, or MIC, of metals and structures, leading to significant maintenance and replacement costs. Number B, of implications of biofilm formation is infection. In medical scenarios, biofilms can form on medical implants, catheters, and other devices, leading to chronic infections that are difficult to treat. Number C, of implications of biofilm formation is environmental impact. Biofilms play a crucial role in biogeochemical cycles and can influence water quality in natural ecosystems. Number 4 is management of biofilm. Controlling biofilm formation often involves prevention strategies, which may include regular cleaning and disinfection, using appropriate chemical agents and mechanical methods to disrupt and remove biofilms. Environmental control, altering conditions that favor biofilm growth, such as reducing moisture or changing pH levels. Understanding biofilm formation is critical in various fields, including microbiology, medicine, and environmental science, as it impacts health, industry, and ecosystem functionality. What is metabolic activity? Metabolic activity refers to the biochemical processes that occur within cells, enabling them to maintain life. These processes involve various chemical reactions that transform substances, allowing cells to grow, reproduce, respond to their environment, and carry out the functions necessary for survival. Metabolic activity can be broadly divided into two categories, anabolism and catabolism. Number 1 is key components of metabolic activity. Number A, of key components of metabolic activity is anabolism. This is the set of metabolic pathways that construct molecules from smaller units. Anabolic reactions typically require energy, which is often derived from the breakdown of ATP, adenosine triphosphate. Examples include protein synthesis from amino acids, DNA replication, synthesis of complex carbohydrates from simpler sugars. Number 2 is measurement of metabolic activity. Metabolic activity can be measured in various ways, often depending on the context. Number A, respiration rates. The rate of oxygen consumption or carbon dioxide production can indicate cellular metabolism, particularly in aerobic organisms. Number B, biochemical markers, enzyme activity, substrate concentration, and the production of byproducts, like lactate or ethanol, can also serve as indicators of metabolic rates. Number C, colorimetric and fluorescent assays. These assays can be used in laboratories to quantify metabolic activity based on the activity of certain enzymes or the utilization of specific substrates. Number 3 is importance of metabolic activity. Number A, of importance of metabolic activity is cellular function. Metabolic activity is essential for maintaining cellular integrity, function, growth, and division. Number B, of importance of metabolic activity is homeostasis. It helps regulate internal environments, homeostasis, by managing energy production, nutrient consumption, and waste elimination. Number C, of importance of metabolic activity is response to environment. Cells can adjust their metabolic activities in response to changes in their environment. For instance, during nutrient scarcity, cells may shift toward catabolism to obtain energy. Number D, of importance of metabolic activity is health and disease. Changes in metabolic activity can be indicative of various diseases or health conditions. For instance, elevated metabolic rates may link to cancer, while reduced activity can indicate issues such as metabolic syndrome. In summary, metabolic activity encompasses all the chemical reactions and processes that support life within a cell, making it a fundamental aspect of biology, physiology, and biochemistry. What is electrochemical reactions? What is electrochemical reactions? Number 1 is key concepts of electrochemical reactions in MIC. Number A, of key concepts of electrochemical reactions in MIC is corrosion basics. Corrosion is an electrochemical process involving the oxidation of metals, where the metal loses electrons, typically in the presence of moisture and an electrolyte, such as salt water. The overall reaction leads to the deterioration of the material. Number B, of key concepts of electrochemical reactions in MIC is role of microorganisms. Certain microorganisms, including bacteria and fungi, can facilitate or exacerbate corrosion through various mechanisms, such as oxidation of metals. some bacteria can oxidize metals directly, contributing to the loss of electrons from the metal surface. Production of corrosive metabolites. Microbes can produce acids, like acetic acid or sulfuric acid, or other metabolites that lower the pH of the surrounding environment, increasing corrosion rates. Number C, of key concepts of electrochemical reactions in MIC is electrochemical mechanisms. In the context of MIC, several important electrochemical reactions can occur, such as anodic reaction. The oxidation of metal occurs at the anode, where metal ions dissolve into the electrolyte. For example, iron can oxidize to form iron ions, Fe superscript 2. Fe superscript 2 Cathodic reaction. Concurrently, a reduction reaction occurs at the cathode, which may involve the reduction of oxygen, in aerobic environments, or other species in anaerobic conditions. For example, oxygen can be reduced to water. O24E-2H2O4OH. Microbially driven cathodic reactions. Certain bacteria, like sulfate-reducing bacteria, SRBs, can facilitate cathodic reactions that are biologically mediated, such as the reduction of sulfate, SO4 superscript 2, to sulfide, S superscript 2. Number D, of key concepts of electrochemical reactions in MIC is biofilms. Many microorganisms involved in MIC form biofilms on metal surfaces. These biofilms can create microenvironments that alter local chemistry, enhancing localized corrosion. Within biofilms, different areas may have varying levels of oxygen, leading to differential aeration corrosion or localized pits. Number 2 is implications of MIC in electrochemical reactions. Number A, of implications of MIC is material degradation. MIC can lead to significant degradation of metals used in pipelines, storage tanks, marine structures, and various industrial equipment, resulting in structural failures, economic losses, and safety hazards. Number B, of implications of MIC is control and prevention. Understanding the electrochemical mechanisms of MIC can help in developing strategies to control or prevent it, such as using biocides or corrosion inhibitors, implementing effective cleaning techniques to remove biofilms, designing materials, such as using corrosion-resistant alloys, that are less susceptible to microbial activity. Understanding the role of electrochemical reactions in microbiologically induced corrosion is crucial for managing and mitigating corrosion-related issues in various industries, particularly those reliant on metal infrastructure. Microbiologically induced corrosion affected materials based on API 571, most common materials of construction including carbon and low alloy steels, 300 series SS and 400 series SS, aluminum, copper and some nickel-base alloys. Microbiologically induced corrosion, MIC, can affect a wide range of materials, particularly those commonly used in industrial settings and infrastructure. The materials most susceptible to MIC damage include: Number 1 is metals. Number A, of metals is carbon steel, one of the most commonly affected metals due to its widespread use in pipelines, tanks, and structures. MIC can cause significant pitting and localized corrosion. Number B, of metals is stainless steel, while generally more resistant to corrosion, certain grades, especially those with lower chromium content, can still be susceptible to MIC under specific conditions. Number C, of metals is cast iron, often used in pipe systems and machinery. Cast iron can suffer from MIC, especially in environments with high levels of moisture and bacteria. Number D, of metals is aluminum and its alloys. Although aluminum has good corrosion resistance, it can still experience MIC, particularly in the presence of aggressive organisms or when protective oxide films are disrupted. Number E, of metals is copper and copper alloys. MIC can occur in copper, particularly in warm, moist environments where biofilms form. Number 2 of the materials most susceptible to MIC damage is concrete and cementitious materials. While concrete is not a metal, it can also be affected by MIC. Microorganisms can colonize surfaces, producing organic acids that can degrade the concrete's integrity, especially in the presence of moisture and nutrients. Number 3 of the materials most susceptible to MIC damage is polymer and composite materials. Number A of polymer and composite materials is some polymer materials, particularly those used in pipelines and tank linings, can be affected by microbial activities. Certain bacteria can metabolize or degrade polymers, leading to material failure over time. Number B of polymer and composite materials is composites can also be susceptible if they contain fillers or additives that are digestible by bacteria. Number 4 of the materials most susceptible to MIC damage is coatings and linings. Protective coatings designed to prevent corrosion can be compromised by MIC if microbes penetrate or degrade these layers. Biocorrosion can occur under coatings if moisture becomes trapped, promoting microbial growth. Number 5 of the materials most susceptible to MIC damage is soils and sediments. Although not a structural material, soils and sediments can influence MIC in buried pipelines and structures. The presence of anaerobic bacteria in these environments can lead to corrosion of buried metal. In summary, MIC affects a variety of materials, with metals like carbon steel and stainless steel being the most commonly impacted. The susceptibility of materials to MIC depends on their composition, environmental conditions, and the presence of specific microbial communities. Understanding the types of materials at risk is essential for developing effective mitigation strategies to prevent MIC damage. Microbiologically Induced Corrosion, or MIC, critical factors based on API 571. Number A, MIC is usually found in aqueous environments or surfaces where water is always or sometimes present, especially where stagnant or low-flow conditions allow and/or promote the growth of microorganisms. Number B, because there are several types, organisms can survive and grow under severe conditions including lack of oxygen, light or dark, high salinity, pH range of 0 to 12, and temperatures from 0 degrees Fahrenheit to 235 degrees Fahrenheit, or minus 17 degrees Celsius to 113 degrees Celsius. Number C, systems may become inoculated by the introduction of organisms that multiply and spread unless controlled. Number C, different organisms thrive on different nutrients including inorganic substances, such as sulfur, ammonia, H2S, and inorganic substances, such as hydrocarbons, organic acids. In addition, all organisms require a source of carbon, nitrogen, and phosphorus for growth. Number E, in leakage of process contaminants such as hydrocarbons or H2S may lead to a massive increase in biofouling and corrosion. Microbiologically induced corrosion, or MIC, is influenced by several critical factors that can exacerbate or mitigate microbial growth and subsequent corrosion processes. Understanding these factors is essential for effective prevention and control. Here are the microbiologically induced corrosion, or MIC, key critical factors. Number 1 is microbial species and community composition. Different microorganisms are associated with MIC, including bacteria, fungi, and archaea. The specific community structure and types of microbes present, such as sulfate-reducing bacteria, SRB, iron-oxidizing bacteria, play a significant role in the formation and severity of corrosion. Number 2 is environmental conditions. pH, the acidity or alkalinity of the environment can significantly influence microbial activity and corrosion rates. Many corrosive microbes thrive in specific pH ranges. Temperature, higher temperatures can accelerate microbial metabolism and increase corrosion rates. Certain bacteria are more active and virulent at elevated temperatures. Oxygen levels, the presence of oxygen can affect the types of bacteria that thrive in a given environment, as some are aerobic while others are anaerobic. Anaerobic conditions often promote corrosion by SRB, for instance. Number 3 of the microbiologically induced corrosion, or MIC, key critical factors is water activity and moisture content. Sufficient moisture is critical for microbial growth and metabolism. Microscopic films of water on surfaces create ideal conditions for biofilm formation and corrosion. Number 4 of the microbiologically induced corrosion, or MIC, key critical factors is nutrient availability. The presence of organic and inorganic nutrients can enhance microbial growth. Common nutrients include nitrogen, phosphorus, and carbon sources, which can often be sourced from spills, leaks, or organic matter in the environment. Number 5 of the microbiologically induced corrosion, or MIC, key critical factors is material composition. The type and mechanical properties of materials significantly influence susceptibility to MIC. Some metals, such as carbon steel, are more vulnerable, while others, such as stainless steel, exhibit greater resistance depending on alloying elements. Number 6 of the microbiologically induced corrosion, or MIC, key critical factors is surface conditions. Surface roughness, cleanliness, and the presence of protective coatings affect the likelihood of biofilm formation. Rough surfaces are more prone to retaining moisture and promoting microbial adhesion. Number 7 of the microbiologically induced corrosion, or MIC, key critical factors is hydraulic conditions. Flow dynamics, such as turbulence and stagnation zones in pipelines or tanks, can influence biofilm formation. Laminar flow may allow for more significant biofilm growth, while high velocity can disrupt established colonies. Number 8 of the microbiologically induced corrosion, or MIC, key critical factors is electrochemical factors. Galvanic couples, where two different metals are in contact, and prevailing electrical potentials can affect localized corrosion processes influenced by microbial activity. Number 9 of the microbiologically induced corrosion, or MIC, key critical factors is presence of biofilms. Biofilms provide a protective environment for microbes, allowing them to thrive and exert corrosive effects. They can also shield corrosive agents from protective materials, complicating mitigation efforts. Number 10 of the microbiologically induced corrosion, or MIC, key critical factors is climate and local environment. External environmental factors, such as humidity, rainfall, and temperature fluctuations, can directly influence MIC by altering moisture levels and microbial activity. In summary, MIC is influenced by a complex interplay of biological, chemical, and physical factors. Identifying and understanding these critical factors is crucial for implementing effective strategies for monitoring, preventing, and managing MIC-related damage in industrial and infrastructure settings. Regular monitoring, material selection, environmental control, and maintenance practices can help mitigate the risks associated with MIC. Microbiologically induced corrosion, or MIC, affected units or equipment based on API 571, Number A, MIC is most often found in heat exchangers, bottom water of storage tanks, piping with stagnant or low flow, and piping in contact with some soils. Number B, MIC is also found in equipment where the hydratist water has not been removed or equipment has been left outside and unprotected. Number C, product storage tanks and water-cooled heat exchangers in any unit where cooling water is not properly treated can be affected. Number D, fire water systems can be affected. Microbiologically induced corrosion or MIC can affect a wide range of units and equipment in various industries, leading to significant operational challenges, safety concerns, and financial losses. Below are the key types of microbiologically induced corrosion or MIC affected units and equipment. Number 1 is pipelines. Oil and gas pipelines. Corrosion can lead to leaks and catastrophic failures in pipelines transporting crude oil, natural gas, and refined products. Water distribution pipelines. Municipal and industrial water supply systems can experience accelerated corrosion due to microbial activity, affecting water quality and system integrity. Chemical transport pipelines. Pipelines carrying corrosive or reactive chemicals may also be vulnerable, leading to safety hazards. Number 2 is storage tanks. Underground storage tanks, USTs common in the petroleum industry, these tanks can corrode due to the presence of water and microorganisms, leading to leaks and environmental contamination. Above-ground storage tanks, ASTs tanks used for storing chemicals, fuels, or wastewater can be compromised by MIC, affecting overall facility safety. Number 3 is process equipment. Heat exchangers. Equipment used for transferring heat can develop biofilms and localized corrosion, reducing efficiency and causing leaks. Pressure vessels. Vessels used in various industrial processes may suffer from MIC, jeopardizing safety and operational reliability. Reactors. Bioreactors or chemical reactors may be affected, especially in processes involving organic substrates that support microbial growth. Number 4 is marine infrastructure. Ship hulls. Microbial activity in marine environments can lead to corrosion of ship hulls, affecting hull integrity and increasing fuel consumption. Offshore platforms. Structures in marine environments, like oil rigs, are particularly susceptible to MIC due to exposure to seawater and organisms. Number 5 is cooling systems. Cooling towers. The warm, moist environment in cooling towers can foster biofilm growth and corrosion in cooling water systems. Condensers and chillers. Heat exchange units in HVAC systems can corrode due to microbial activity. Number 6 is wastewater treatment facilities. Digesters. Anaerobic digesters can be impacted by MIC, affecting the efficiency of organic material breakdown and subsequent sludge management. Clarifiers and tanks. Equipment for settling and separating solids can experience increased corrosion rates due to microbial interactions. Number 7 is industrial equipment. Pumps and valves. Components used to move fluids can face corrosion issues, leading to leaks and equipment failures. Rotary equipment. Equipment such as agitators or mixers can corrode due to corrosive environments fostered by MIC. Number 8 is food processing equipment. Processing machinery. Machinery that processes food can suffer from MIC, impacting product safety and equipment longevity. Storage containers. Tanks and vessels used for food storage, especially if moisture-laden, can corrode. Number 9 is concrete structures. Reinforced concrete elements. Some microorganisms can degrade concrete and corrode embedded steel reinforcement, impacting structural integrity, particularly in sewage treatment plants or marine structures. Implications of MIC. Cross-industry implications include: Number 1 is safety hazards. Increased risk of leaks, structural failures, and environmental contamination. Number 2 is maintenance costs, higher maintenance and repair costs due to accelerated wear and failure. Number 3 is operational downtime, disruptions in processes and operations leading to loss of productivity. Number 4 is regulatory compliance, possible violations of environmental regulations leading to fines and penalties. By understanding the units and equipment affected by MIC, industries can better implement preventive measures and maintain asset integrity. Appearance or morphology of microbiologically induced corrosion damage based on API 571. Number A. MIC corrosion is usually observed as localized pitting under deposits or tubercles that shield the organisms. Number B. Damage is often characterized by cup-shaped pits within pits in carbon steel or subsurface cavities in stainless steel. See Figure 489, MIC of diesel tank bottom. See Figure 490, pitting corrosion on the ID of a 6-inch CS sour crude line after 2.5 years of service. Pits are approximately 1 inch to 2 inch wide. Note the halo effect in Figure 491. See Figure 491, same pipe as Figure 490. Note the halo effect. See Figure 492, oil line with MIC damage beneath tubercles. See Figure 493, same oil line as Figure 492. Hemispherical pitting typical of MIC can be seen after grit blasting to remove the scale. See Figure 494, Type 304 stainless steel exchanger tubes failed from pitting corrosion on the shell side in cooling water service after 2.5 years without biocide treatment. See Figure 495, a cross-section of the tube, Figure 494, revealing severe subsurface tunneling, typical of MIC. Microbiologically induced corrosion or MIC damage exhibits specific appearances and morphologies that can help in identifying its occurrence. Here are some key characteristics and forms in which MIC damage commonly manifests. Number 1 is localized corrosion. Pitting corrosion. Small, localized pits that penetrate the metal surface deeply. Pitting is often observed due to the localized attack by sulfate-reducing bacteria, SRBs, and other corrosive microorganisms. Crevice corrosion occurs in areas where there are tight spaces, such as joints or under deposits, that can trap moisture and nutrients, facilitating microbial growth and localized corrosion. Number 2 is surface biofilms. Biofilm formation, a slimy layer of microorganisms that adhere to metal surfaces. The biofilm may appear as a translucent, greenish, or blackish layer depending on the type of microorganisms present. Structure: these biofilms can trap sediments and promote anaerobic conditions beneath them, further enhancing corrosion processes. Number 3 is galvanic corrosion features. Anodic and cathodic areas, mic may lead to corrosion patterns that resemble galvanic corrosion, where certain areas corrode preferentially due to differing galvanic potentials induced by microbial activity. Number 4 is rust and stains. Color changes: corroded areas, particularly on iron-based alloys, may exhibit reddish-brown rust-colored stains or patches, indicating iron oxide formation. Discoloration: non-ferrous metals can show discoloration or surface staining due to microbial activity, which can also affect protective coatings. Number 5 is filiform corrosion. Filiform patterns: this type of corrosion occurs beneath a surface coating, like paint, and manifests as thread-like filaments of corrosion that can extend across surfaces, often caused by microbial activity. Number 6 is erosion corrosion. Surface flaking or peeling: in scenarios where microbial activity combines with fluid motion, like in cooling towers or pipes, the surfaces may flake or peel, indicating erosion corrosion processes. Number 7 is other morphological features. Tuberculation: the formation of small, tubercle-like projections on surfaces, commonly seen on steel or metal components exposed to certain water conditions, often due to biofilms and anaerobic bacteria. Corrosion patches: discrete areas of corrosion that may vary in size and depth, often surrounded by unaffected or less affected areas of the substrate. Number 8 is under-deposit corrosion. Corrosion beneath deposits: when sediment or debris accumulates on a metal surface, microbes can thrive underneath, leading to localized corrosion that is often difficult to detect until significant damage occurs. In summary, the appearance and morphology of MIC damage can vary widely based on environmental conditions, the specific types of microorganisms involved, and the materials of the affected equipment. Diagnostic techniques like visual inspections, microscopy, and chemical analyses may be employed to confirm MIC and assess its severity and extent on affected structures. Identifying these characteristics early can help prevent extensive damage and reduce maintenance costs. Prevention and mitigation of microbiologically induced corrosion or MIC based on API 571, Number A, microbes require water to thrive. Systems that contain water, cooling water, storage tanks, etc., should be treated with biocides such as chlorine, bromine, ozone, ultraviolet light or proprietary compounds. Number B, proper application of biocides will control but not eliminate microbes so that continued treatment is necessary. Number C, maintain flow velocities above minimum levels. Minimize low flow or stagnant zones. Number D, systems that are not designed or intended for water containment should be kept clean and dry. Number E, empty hydratist water as soon as possible. Blow dry and prevent moisture intrusion. Number F, wrapping and cathodically protecting underground structures have been effective in preventing MIC. Number G, effective mitigation of established organisms requires complete removal of deposits and organisms using a combination of pigging, blasting, chemical cleaning and biocide treatment. Number H, add biocides to water phase in storage tanks. Number I, maintain coatings on the interior of storage tanks. Preventing and mitigating microbiologically induced corrosion, or MIC, involves a combination of strategies that target microbial growth and the environmental conditions conducive to corrosion. Here are several effective measures to combat MIC damage. Number 1 is regular monitoring and inspection. Corrosion monitoring: utilize sensors and corrosion monitoring systems to detect early signs of corrosion. Regularly inspect pipelines, tanks, and equipment for initial signs of microbial activity and corrosion. Microbial testing. Conduct microbiological testing to identify the presence and types of microorganisms involved in MIC. This helps in tailoring mitigation strategies. Number 2 is cleaning and maintenance. Routine cleaning. Regularly clean surfaces to remove biofilms, sediments, and other deposits that can harbor microbial growth. Methods can include chemical cleaning agents, high-pressure water jets, or mechanical scraping. Maintenance schedule. Implement a systematic maintenance schedule that includes equipment inspections and cleaning to prevent buildup and corrosion. Number 3 is chemical treatments. Biocides. Use biocidal chemicals to reduce or eliminate microbial populations. The selection of biocides should depend on the system and potential impact on operations. Common options include chlorination, bromination, or quaternary ammonium compounds. Corrosion inhibitors. Apply corrosion inhibitors that can help minimize the corrosive effects of any microbial activity that is present. These substances can coat metal surfaces, making them less susceptible to corrosion. Number 4 is control of operating conditions. Reduce nutrients. Lower the availability of nutrients, like organic matter, that support microbial growth. This can involve controlling the chemical composition of water or processing fluids. Temperature control. Maintain optimal temperatures for systems, as elevated temperatures can promote microbial growth. Depending on the application, this may involve reducing water temperatures or monitoring heat management systems in reactors or bioreactors. Number 5 is material selection and coating. Corrosion-resistant materials. Use materials that are less susceptible to MIC, such as stainless steel or non-ferrous alloys, in environments prone to microbial attacks. Protective coatings. Apply anti-corrosion coatings or linings to metal surfaces. Specialized coatings can provide a barrier to both corrosion and microbial attachment. Number 6 is system design modifications. Avoid stagnation. Redesign systems to minimize stagnant areas where microbes can proliferate. Ensure proper flow dynamics in pipelines, tanks, and cooling systems. Effective drainage. Implement drainage systems to prevent standing water or trapped moisture in containers and equipment that are vulnerable to MIC. Number 7 is education and training. Staff training. Train personnel on the importance of MIC and the measures that can be taken to prevent it. Awareness can enhance compliance with maintenance procedures and inspection protocols. Best practices implementation. Develop and implement best practices for monitoring microbiological activity and corrosion within each specific operation. Number 8 is collaborative research and development. Industry collaboration. Participate in research initiatives to study MIC and develop new technologies or approaches for its prevention. Feedback loops. Evaluate the effectiveness of conducted strategies and share results within and among industries to improve understanding and mitigation of MIC. In conclusion, preventing and mitigating MIC requires a multifaceted approach that combines routine monitoring, effective chemical treatments, systematic maintenance, material selection, and design considerations. By proactively implementing these strategies, industries can significantly reduce the risks associated with microbiologically induced corrosion and preserve the integrity of their equipment and infrastructure. Microbiologically induced corrosion inspection and monitoring based on API 571, Number A, in cooling water systems, effectiveness of treatment is monitored by measuring biocide residual, microbe counts and visual appearance. Special probes have been designed to monitor for evidence of fouling which may precede or coincide with MIC damage. Number C, an increase in the loss of duty of a heat exchanger may be indicative of fouling and potential MIC damage. Number D, foul-smelling water may be a sign of trouble. Detecting microbiologically induced corrosion, or MIC, involves several inspection and monitoring methods aimed at identifying microbial activity, assessing corrosion damage, and understanding the environmental conditions contributing to MIC. Here are some commonly used of inspection and monitoring methods. Number 1 is visual inspection. Surface examination. Conduct routine visual inspections of equipment, pipelines, tanks, and other structures for signs of corrosion, such as pitting, rust, discoloration, or biofilm formation. Use of borescopes. Employ borescopes or video cameras for visual inspection of internal surfaces that are difficult to access, allowing for inspection of crevices and confined spaces. Number 2 is microscopic examination. Scanning electron microscopy, SEM high-resolution imaging to examine surface morphology and to identify microbial biofilms and corrosion products on metal surfaces. Optical microscopy, used for preliminary examinations of surfaces to identify and characterize biofilms, corrosion pits, or other factors indicative of MIC. Number 3 is microbiological sampling. Water and biofilm sampling. Collect water samples or biofilm specimens from affected surfaces to perform microbiological assays specific to the types of bacteria associated with MIC, such as sulfate-reducing bacteria, iron-oxidizing bacteria. Culturing techniques. Employ selective culture methods that help in isolating and quantifying specific microbial species capable of causing corrosion. Number 4 is corrosion rate measurement. Weight loss coupons. Install corrosion coupons made from the same material as the system components. Measure weight loss over time to estimate corrosion rates, which can indicate MIC activity. Electrochemical techniques. Use techniques like linear polarization resistance, LPR, or electrochemical impedance spectroscopy, EIS, to assess corrosion rates in real time based on electrochemical measurements. Number 5 is nondestructive testing, or NDT. Ultrasonic testing, or UT, measure the thickness of the material to detect reduced thickness due to corrosion. This can indicate localized corrosion events typical in MIC. Radiography. Utilize X-rays or gamma rays to inspect subsurface structures for signs of corrosion or structural integrity without causing damage. Number 6 is field testing for specific microorganisms. Biochemical assays. Perform rapid field tests to measure specific metabolites associated with MIC, such as hydrogen sulfide, produced by sulfate-reducing bacteria. This can help gauge microbial activity. DNA or RNA testing. Use polymerase chain reaction, PCR, or other molecular biology techniques to identify and quantify specific microbial communities present in water or biofilms. Number 7 is environmental monitoring. Chemical analysis. Test water chemistry for parameters such as pH, oxygen levels, and nutrient concentrations, like sulfates, nitrates. These factors can influence microbial growth and MIC activity. Temperature and flow rate monitoring. Continuous monitoring of temperature and fluid flow rates in pipelines or storage tanks, as fluctuations can impact microbial growth and corrosion rates. Number 8 is corrosion mapping. 3D mapping technologies. Utilize advanced mapping techniques, including drone-assisted imaging or laser scanning, to create 3D maps of surfaces, helping to identify areas that may be more susceptible to MIC based on their morphology and corrosion rates. In conclusion, using a combination of these inspection and monitoring methods enables a comprehensive understanding of MIC risks and facilitates timely intervention. Regular monitoring and analysis of microbial activity and corrosion rates are critical for preventing extensive damage and ensuring the longevity of materials and infrastructure in susceptible environments. Thank you.